So I want you to imagine that you're a nightclub bouncer, and your job is, well, it's terrifyingly specific, right?
Mm-hmm.
You have to strictly alternate letting people into the club and then kicking them out exactly every 90 minutes.
Right. And if you freeze up and just leave the door wide open, the club catches fire and burns to the ground.
Exactly. But, if you panic and keep the door permanently shut, the club goes completely bankrupt. So you have to cycle between open and closed perfectly over and over with literally zero margin for error. And that life or death scenario is exactly the environment scientists created for microscopic proteins inside living yeast cells. So welcome to today's deep dive. We're completely unpacking a fascinating 2026 paper published in the journal Cell by Gligorovsky and colleagues. It's all about this concept called optovolution. And our mission today is to figure out exactly how these researchers hacked the biological clock of a yeast cell to create this ruthless, light-controlled boot camp for evolving, dynamic, shape-shifting proteins.
Which is, it's a massive leap forward. Because historically, when we talk about directed evolution, the process of forcing proteins to evolve new traits in a lab, we've really only been good at making static proteins.
Like things that are just always on or always off.
Exactly, yeah. We can easily select for a protein that binds to a target with incredible strength, or one that glows the absolute brightest green. But if we want to build biological computers, we need proteins that make dynamic decisions. We need logic gates. We need switches that can actually toggle.
Right, because a computer isn't just a wire that's always conducting electricity. It's a series of microscopic switches turning on and off in these incredibly complex patterns.
Mm-hmm.
But if, directed evolution usually just breeds proteins that get stuck in the on position, how do you even begin to breed a protein that knows when to let go?
Well, that's the core problem they're tackling here. To evolve a dynamic switch, you have to apply this intense evolutionary pressure to literally every state of that protein. You have to force it to turn on, force it to turn off, and force the transition between those two states to be completely flawless. Because
if you only reward a protein for turning on, it'll just quickly mutate to stay on forever, right? It just entirely loses the ability to reset.
Right. The bouncer leaves the door open and the building burns down.
Yeah.
So to build this biological boot camp to force that continuous switching, the researchers took budding yeast and engineered it so that a new, unproven protein, we can call it the target protein, directly controls the yeast's own cell cycle.
Okay, wait, so how did they do that? Because I know they hooked this target protein up to two critical cell cycle regulators, the PhyB-PIF and CLN2 cyclins, right?
Yeah, the PhyB-PIF and CLN2 cyclins. To understand why this works, think of the yeast cell cycle as a highly rigid manufacturing assembly line. PhyB-PIF is basically the foreman in charge of the DNA copying phase. The cell absolutely cannot divide without it. But, and here's the massive biological trap the researchers utilized, PhyB-PIF is essentially poisonous to the cell if it stays active during the next phase of the assembly line.
Oh man, okay. So it's like the starter motor in a car engine. You desperately need it to turn the engine over, but the absolute second the engine catches, you have to disengage that starter motor. Because if you leave it engaged while you're, driving down the highway, the whole mechanism just rips itself apart.
That is exactly how it works physically inside the cell. The target protein driving this process must pulse on to start the division, and then it literally must turn off to let the cell finish dividing. If the protein fails to switch, the cell skips essential cycles, the internal machinery breaks down, and the yeast dies.
It's just a brutal pass-fail test.
It really is. They linked the protein's ability to physically toggle back and forth directly to the organism's survival. And to dictate exactly when the protein should toggle, they used optogenetics. They use pulsing light.
I love that because light is like the ultimate precision tool, right? You can flash it on and off in a fraction of a second, completely bypassing the yeast's natural 90-minute internal clock. So I like to visualize this as a flashing traffic light inside the cell. Green means the cell cycle advances, but if the light gets stuck on green, the cell dies. It has to cycle to red and back to green.
That's a great visual memory tool. The protein has to obey the traffic light perfectly.
But wait, I have to push back on the underlying logic here for a second. Yeast cells have been surviving and dividing for over 1 billion years. They have their own robust, deeply ingrained backup systems to manage their cell cycles. So how on earth do you force a yeast cell to just hand over the steering wheel to some new, unproven, light-activated protein without the yeast's natural autopilot just overriding it?
Well, you have to physically rip the autopilot out of the dashboard. The researchers obviously couldn't use normal yeast, so they created these custom, heavily modified strains. They named them the Dodgebog strains. specifically strains like Dodgebog 1 and Dodgebog 2.
Okay, and what did they actually do to these Dodgebog strains?
They went into the yeast's genome and completely deleted the natural cyclins and the natural inhibitors. They used SIC1 delta mutations to strip the cell of its innate ability to regulate its own division. So its survival became 100% dependent on the artificial light-driven cycle.
But hold on, if you take a yeast cell, rip out its natural ability to divide, and replace it with a brand new, unmutated light switch protein that probably barely works yet, I mean, wouldn't the yeast just die immediately? How do you even get the boot camp started if all your recruits drop dead on day one?
So you give them a temporary life support system. The researchers introduced what they call a training wheel plasmid. It's a small, separate loop of DNA carrying backup copies of those essential division genes.
Oh, that's clever.
Right. This keeps the cells alive and dividing normally, while the population grows in the petri dish. Then, when the scientists are ready to begin the actual brutal selection process, they introduce a specific chemical into the environment. And this chemical selectively targets and destroys that backup training wheel plasmid.
Wow, okay. So the life support is violently unplugged, and now it's sink or swim based purely on this experimental light switch protein.
Exactly. And to actually give the yeast a fighting chance to evolve a solution before it dies, the scientists intentionally sabotage the yeast's DNA replication machinery. Normally, when DNA copies itself, there's a molecular spell checker that fixes typos. They introduce the POL3L523D mutation, which essentially blinds that spell checker.
So the mutation rate just absolutely skyrocketed.
Yeah, it rockets up by about 100-fold. And if that wasn't enough, they even blasted the petri dishes with ultraviolet light right before the trial started, just to scramble the genetic code even further.
They just plunge the yeast into a life-or-death crisis, kick away their safety net, and bombard them with UV radiation to force rapid, chaotic mutation. That's intense.
Yeah.
Okay, so we're about 5 minutes in. Let's do a quick recap of what we've unpacked so far. Bullet 1, optovolution works by wiring a protein's on-off switch directly to cell survival. Bullet 2, they use light to force the protein through its paces, making it act like a flawless biological bouncer.
Perfect summary.
So a question for you listening right now. Have you ever had to learn a highly complex new skill under an intense, literal ticking clock deadline? Because that is exactly the environment these yeast cells are placed in. Which brings us to our next big question.
Yeah.
What happens when the light show actually starts? What proteins did they throw into this crucible?
So the first test subject was a very popular optogenetic tool called L222. It's a LV transcription factor. And LV stands for light oxygen voltage. At a molecular level, you can imagine it as a microscopic cage holding a tiny antenna molecule.
Okay, a cage with an antenna.
Yeah. And when a photon of blue light hits that antenna, the energy causes the cage to physically change shape. It exposes this sticky segment that grabs onto DNA and turns a specific germ on.
And then I assume when the light goes away, the cage cools down, folds back up, and releases the DNA.
Exactly. That's the baseline mechanic. But the researchers wanted to push it to its absolute physical limits. So in their first campaign, they wanted to evolve a version of this protein that was hypersensitive to light. To do that, they drastically changed the pulsing rhythm. Instead of generous amounts of light, they flashed the blue light for just 1 single second, followed by 100 minutes of total darkness.
Wait, 1 second of light every hour and 40 minutes? I mean, that is practically pitch black. The baseline protein wouldn't catch nearly enough photons to trigger the cell division, so the baseline yeast would just die, right? How does a protein physically adapt to survive that kind of starvation?
Well, through that accelerated mutation process we talked about, the surviving yeast cells produce proteins with altered physical architectures, mutants like A157V and E84D. These mutations physically tweak the shape of that light-catching pocket, making it wider, or making the cage much more eager to spring open at the slightest introduction of energy.
That's amazing.
It is. The successful strong LOV mutants could produce transcription levels up to 16 times higher than the original protein under incredibly dim conditions.
That's a massive optimization. But, biology always demands a trade-off. If you engineer a mousetrap to have a hair trigger so it snaps at the slightest breeze, it's probably going to be incredibly difficult to pry back open.
You nailed it. That is exactly what happened. The trade-off for this hypersensitivity was a drastic loss of elasticity. Because the mutated protein was so eager to spring open and grab the DNA, it took much, much longer to let go once that brief flash of light was gone. So the dark state recovery became incredibly slow.
Which means it's bordering on staying stuck in the on position. going back to our earlier engine analogy, the startle motor is sticking. So if hypersensitivity creates a sticky, leaky protein that wants to stay on, how do you force the evolution in the opposite direction? How do you actually fix a leaky switch?
You change the punishment mechanism. So in the next campaign, campaign 2, they used long, generous pulses of light, 30 seconds on, 30 seconds off. But, and this is the genius part, they flooded the environment with a constant, very dim background light.
Oh, wow. That is a brilliant trap. Because the background light is essentially static noise. So if the protein is too sensitive or too leaky, it'll interpret that dim background noise as a real signal to turn on. It'll trigger the color-5 cyclin during the dark phase, poison the cell, and die.
Precisely. To survive this specific environment, the protein must physically tighten its structure. It has to become rigid enough to ignore the constant low-level static, but still flexible enough to snap open when the intense 30-second pulse hits. And the survivors of this campaign, specifically the D31Y mutant, developed into incredibly tight switches. The leakiness levels practically dropped to absolute zero.
It's just fascinating how merely changing the pattern and intensity of the light completely dictates the physical architecture of the surviving proteins. But sensitivity and leakiness are really just refining what the protein already does. The paper describes a third campaign, a spectral shift, that completely shatters what we thought these proteins were capable of.
Oh yeah, the impossible shift. So L222, as we mentioned, is specifically built to catch blue light. Blue photons carry a very specific amount of energy, and the physical cage of the protein is evolutionarily tuned to that exact wavelength. For decades, the consensus in the synthetic biology community was that you could not fundamentally change the color LV domain responds to without just entirely breaking the protein's delicate internal mechanics.
Right. It would be like trying to train a dog to respond to a dog whistle that is physically outside of its hearing range. You can't just will the biology to perceive a different frequency. So the researchers take this blue light protein, stick it in the boot camp, and just start flashing green light instead.
Right.
By all biological logic, the protein shouldn't even register the green light, meaning it never turns on, meaning the cell dies.
But the relentless pressure of optovolution found a way. The accelerated mutation-generated variants, specifically the T83A and G80R mutants, that successfully responded to green light. The amino acid sequence shifted just enough to alter how the internal antenna absorbed energy, allowing it to actually utilize the lower energy green photons.
But wait, I have to ask, how efficient could that possibly be? If you're radically redesigning a protein's fundamental energy source on the fly, it can't operate smoothly. Does the green light mutant actually work well?
This is where we see this really stark difference between studying biology in a test tube versus studying it inside a living, messy cell. When scientists extracted one of these green light mutants, like T83A, and tested it in a sterile laboratory solution, what we call in vitro, it looks like a complete failure.
Really? How bad was it?
Its dark recovery was agonizingly slow. After sitting in the dark for 16 hours, the protein had only reverted back to 20% of its original state. So by any standard biochemical metric, the protein was fundamentally broken.
But wait, the cell cycle is only 90 minutes long? If the protein takes 16 hours just to partially reset, how could the yeast cell possibly be surviving the boot camp? The math doesn't add up at all.
Well, it doesn't add up in a vacuum, but living cells are dynamic systems. Inside the living yeast in vivo, the researchers used mathematical modeling and single-cell fluorescence tracking to watch the mechanics in real time. And they discovered that the protein didn't actually need to reset completely. As the green light pulsed, the protein managed this tiny, sluggish oscillation. The difference between its on state and its off state was only about 3.8%.
A 3% ripple, that's it.
That's it. But that microscopic ripple was just enough to trigger the cyclin cascade. It was just enough to push the cell over the threshold into the next phase of division without poisoning it. It totally proves that biology doesn't care about our sterile laboratory metrics of perfection. Biology only cares about what is, good enough to survive.
That is a staggering realization. The boot camp doesn't care if the push-up is pretty, it just cares that you get off the floor. But why does this specific achievement matter so much? Like, why is the scientific community so desperate for a green light LOV protein in the first place?
It unlocks color-multiplexing. Imagine trying to fly a drone, but your remote control operates on the exact same radio frequency as your neighbor's garage door. Every time you try to turn the drone left, their garage opens. That's crosstalk.
Oh, I see.
Yeah, previously, if we wanted to put two different light-controlled circuits into a single cell, we couldn't use two LOV domains because shining blue light would just activate both of them at the same time in this chaotic mess.
But now you have one circuit tuned to blue and one circuit tuned to green. You can fly the drone and open the garage totally independently using the exact same underlying protein architecture. It allows for infinitely more complex programming within a single living cell.
Exactly. We're stacking logic gates without crosstalk.
Okay, let's pause here for our second recap. Bullet 1. We learned we can force proteins to evolve extreme sensitivity or completely eliminate leakiness based on the light pattern we use. Bullet 2. Optovolution achieved the impossible by shifting a blue light protein to see green, which unlocks color multiplexing. So listener, think about your own toolkit. What if you could rewire your standard processes to respond to completely unexpected triggers? Because next we're going to see how the yeast cells themselves actually outsmarted the scientists.
Yeah, this next part is wild, because while changing the color spectrum of a protein is an incredible feat of molecular engineering, the next campaign reveals something even more profound about the nature of evolutionary pressure itself. Sometimes when you demand an upgrade, the organism doesn't just change the specific protein you're targeting, it rewrites its entire surrounding infrastructure.
Okay, set the stage for this. What was the hurdle that required an entire infrastructure rewrite?
They shifted away from the LOV domain and looked at a different optogenetic system called PhyB-PIF. Now, this is a system that responds to red and far red light, but it has a massive logistical flaw when you try to use it in yeast. The PhyB protein is essentially blind on its own. It requires an external chemical called a chromophore to actually catch the light. And the specific chemical it needs is called PCB.
So the protein is just the radio casing, but the PCB chemical is the actual metal antenna. If you don't have the antenna, you just can't catch the signal.
Right. And yeast does not naturally produce PCB. So for this system to work, scientists have to continuously purchase this expensive temperature-sensitive chemical and manually feed it to the yeast in the petri dish. It's a huge pain. So the researchers wanted to know, could optovolution force the system to function without this external chemical supply chain?
Wait, they just drop the yeast into the boot camp, pulse the red light, but they completely starve the yeast of the PCB chemical. Now, the yeast is deaf and blind to the light. It can't trigger the cell cycle, but you can't just magically evolve an antenna out of nowhere, right?
You can't. The protein itself didn't suddenly learn to see without a chromophore. Instead, the continuous life or death pressure forced a mutation in an entirely different part of the yeast's genome. The surviving cells had a deletion in a gene called UR1.
Okay, what does the UR1 gene do? How does breaking that gene save the cell?
So UR1 builds what is known as an ABC transporter. You can visualize it as a molecular garbage pump sitting on the cell membrane. Its entire job is to identify toxins and waste products inside the cell and physically pump them outside. Now, it turns out that yeast naturally produces a byproduct molecule called biliverdin, which happens to be chemically very similar to the PCB antenna.
Oh, I see where this is going.
Right. But under normal circumstances, the YR1 garbage pump recognizes biliverdin as useless waste and relentlessly pumps it out of the cell before it can be used.
I absolutely love the mechanics of this. It's like a high-end restaurant chef who thinks they have to import incredibly expensive foreign saffron to make their signature dish, but they're failing because the shipments stopped coming in. Then they realize there's a perfectly viable local substitute weed growing right behind the restaurant. The only problem is the janitor keeps pulling the weeds and throwing them in the dumpster. So the chef doesn't invent a new spice, they just fire the janitor.
That is a perfect way to visualize it. By deleting the OR1 gene, the garbage pump was destroyed. The janitor was fired. So the natural biliverdin stopped being thrown away. It pooled up inside the cell, and the blind PhyB protein just grabbed it, used it as a makeshift antenna, caught the red light, and saved the cell.
That's incredible.
It really is. The evolution solved a systemic supply chain issue by simply removing a barrier. The scientists never intended to mutate the waste pump. The biological algorithm just found the path of least resistance to survival.
Which totally proves that when you tie a mechanism directly to cell survival, the whole organism becomes a canvas for innovation. The yeast is using its own wisdom. But, everything we've talked about so far relies on light. It's optovolution. Is this technology strictly limited to light-activated proteins, or can we use this cell cycle boot camp to evolve other types of biological machinery?
The researchers proved it's broadly applicable by evolving a purely chemical logic gate, too. They took a transcription factor called PSRTA that is normally activated by a chemical called doxycycline. But they wanted to make it more complex. They wanted to create a biological AND gate.
Right, so in computer science, an AND gate means a circuit requires two distinct inputs to fire. Input A and input B must both be present. If you only have one, nothing happens.
Exactly. To build this mechanically in the cell, they fused the transcription factor to something called a degron. A degron is essentially a molecular self-destruct tag. When the cell's natural garbage disposal system sees a degron, it immediately shreds the attached protein. But the chemical input, doxycycline, acts as a shield that hides the self-destruct tag.
So you need the chemical shield to prevent the protein from being destroyed, and you need a separate signal to actually tell the protein to go to work. Two inputs.
Right. And they hooked this chemical AND gate up to the cell cycle survival trap. To provide the second input, they used incredibly short, 2-second pulses of blue light. Because the light pulses were so short, the initial unmutated AND gate was just too weak. The physical bond between the chemical shield and the protein wasn't strong enough to sustain the rigorous 60-minute pulsing requirement. The cells were just dying.
But the boot camp forces an adaptation, obviously.
It did. The evolutionary pressure quickly forged a mutation in the chemical receptor, the F67C mutation. It physically altered the binding pocket so that the doxycycline shield locked in with far greater affinity. It created a much stronger, highly dynamic logic gate that perfectly obeyed the complex two-input requirement, but functioned efficiently enough to keep the cell dividing.
It just proves that we aren't just building better light switches anymore. We are evolving true biological microprocessors. We can force cells to perform complex chemical computations just to stay alive. Okay, time for our final 5-minute recap. Bullet 1, cells can evolve systemic workarounds like deleting the YOR1 gene to save on chemical supply chains. Bullet 2, optovolution works for chemical and computational logic gates, not just light sensors. So listener, what stands out to you about the yeast finding a completely different solution than the scientists expected? Because sometimes the best upgrades really do come from removing a barrier rather than adding a feature.
Yeah, if we step back and look at the broader implications, this methodology completely rewrites the rules of synthetic biology. We are no longer limited to designing static tools in a petri dish. By directly coupling a protein's input-output loop to the ultimate ticking clock of life, the cell cycle, we have unlocked a mechanism for continuous dynamic evolution. We can harness the brutal efficiency of natural selection to calculate and refine logic gates on a molecular scale.
We are literally hacking the fundamental pulse of life to do our computational bidding, which honestly leaves us with a highly provocative thought to chew on long after this deep dive ends. If we can use this pulsing selection pressure to force yeast cells to evolve incredibly complex dynamic logic gates, what happens when we apply this to human biology? Could we eventually use a similar cyclical pressure to evolve synthetic immune cells? I mean, imagine an engineered white blood cell that doesn't just blindly attack. Imagine an immune cell that dynamically learns to recognize a specific cancer signature, computes the exact moment to unleash a lethal attack, and then crucially automatically switches off the absolute second the threat is neutralized to prevent autoimmune damage. We wouldn't just be administering medicine, we would be deploying microscopic, evolving computers inside the human body, adapting their own internal logic on the fly. Thank you for joining us on this deep dive. Keep questioning the mechanics of the world around you, and we'll see you next time.